All right, and welcome to DNA replication, part 2. As I was just saying in part 1, DNA polymerases are enzymes that catalyze a new strand of DNA, but they can't start a strand. So they require a primer, which is laid down by an RNA polymerase called primase. Puts down a little bit of RNA, and then the DNA polymerase can come and read the DNA template strand and catalyze the synthesis of the second strand. Elongation of the strand is very fast in prokaryotes, about 500 nucleotides per second, and much slower in eukaryotes, 50 nucleotides per second. That's because of the presence of histones in eukaryotes that fold the DNA up. We have to get those out of the way, replicate the DNA, and then put them back on. So this is just kind of showing here again, DNA polymerase needs that free 3' hydroxyl group. I don't know why, there we go, 3' hydroxyl group. In order to add the next base, the base comes in, base pairs with its complementary pair, and then DNA polymerase catalyzes the formation of our phosphodiester bond. And then we have another free 3' hydroxyl. We can bring the next base in, which here would be a guanine. DNA polymerase catalyzes the formation of the bond, etc., and so on until they've copied the whole strand. Now, the anti-parallel structure of the double helix affects replication. Okay, so polymerases only add to a free 3' end, so the growing strand elongates in a 5' to 3' direction. Okay, so the template will be, oops, that's totally wrong, in the 5' to 3' direction like this. Okay, and this would be my growing strand. All right, along one template strand of DNA, there is one of the synthesis strands being synthesized is called the leading strand. And the leading strand is moving in the same direction as the replication fork. So if I have a replication fork here, okay, this top is 5' to 3', so that would make this 5' to 3'. Okay, so if I'm making a new strand, let's go purple. Okay, it needs to be made in the 5' to 3' direction. Okay, and the strand that moves in the same direction as the fork, this strand, is called the leading strand, and it is synthesized continuously. Okay, so here is, so we have our origin here, each side is a fork. Okay, so this is where replication starts. So you'll have the leading strand moving the same direction as the fork, and it's made continuously. On the other side of the fork, this would be the leading strand, and it's moving, again, same direction as the fork, and made continuously. And then the fork moves this way and this way. Okay, so here we have everybody. Here we have helicase, and opening up the strands, single-stranded binding protein stabilizing. We have our little primer laid down by our primase. And then here is DNA polymerase. Okay, and you'll see as the fork continues to move in this direction and open up the strand, DNA polymerase continuously synthesizes the leading strand in the same direction as the replication fork. Now, the other strand is a little bit more complicated, okay? It's called the lagging strand. And because DNA, again, can only be made in a 5' to 3' direction, one strand is going to go towards the fork, the other strand is going to have to go away from the fork, okay? So the lagging strand is actually made in a series of little segments called Okazaki fragments. Okay, so again, we had our, I'm still doing purple. So my leading strand here is going towards the fork, 5' to 3', so it's being made 5' to 3'. This is 3' to 5'. So let's go back to red. The bottom strand here, in order to make it go 5' to 3', it has to be made in that direction. So it's made in these little chunks because we have to wait for the strands to separate. Then DNA polymerase can sit down and make another little chunk. Okay, so let's look at that on here. So again, here's 1 Okazaki fragment, 2 Okazaki fragments, 3 Okazaki fragments. As the strands open up a little bit more, you'll get another one. Strands open up a little bit more, you'll get another one. Okay, and this kind of shows the how that works. We've laid down our primer here. Okay, and then RNA polymerase can come and make the rest of the fragment until it meets up with the previous fragment. Then the template has opened up a little bit more in this section here. We get another primer, and DNA polymerase lays down another bit, and so we have a couple of these little Okazaki fragments. Here's one Okazaki fragment, here's another one. Then what happens is we need to get rid of the DNA. So DNA polymerase can cut out the RNA and replace it, because now it has a 3' end to work with, can cut out the RNA and replace it with DNA. And then there's a, so this is DNA polymerase. And then there's an enzyme called DNA ligase that ligates the ends. See this little break here between the two fragments? It fixes that break and makes everybody happy. Okay, so this is kind of a, oh, and just so I get back here, the lagging strand is made discontinuously. That's not even spelled right, discontinuously, away from the fork. So in the opposite direction. So these guys are made away from the fork. Leading strand is made towards the fork. Okay, and then on the other side, leading strand made towards the fork, lagging strand made away from the fork. So here's both strands all together. Here's the leading strand, and here is the lagging strand. Okay, so, and again, polymerase can, DNA polymerase 3 is the main one that makes DNA. DNA polymerase 1 can chop out the RNA and replace it with DNA. And then DNA ligase fixes the nicks. When you have a little break in the backbone, it's called a nick. Okay. Now, all of these enzymes, we show them kind of broken up along a linear stretch of DNA here. There's all these guys kind of in their different spots. And we do that just so you can kind of see where it is. But they really don't function that way. So all these proteins form a very large complex, and we call it the DNA replication machine. And the cool thing is, and there's a video on the thing link that kind of explains this a little bit, but the replication machine sort of stands still, and the DNA kind of gets reeled in or threaded through the replication machine, leading and lagging strands together, and the newly made daughter DNA get kind of extruded out the back. So this kind of shows you a little bit more how it works. Okay, so here you'll see these guys are all held together into one big enzyme. Okay, this is the leading strand right here. It continues on this way, and you'll see the lagging strand has to loop around in order to get the primer up next to the DNA polymerase. Then once that elongates to the end of this primer, okay, it will let go and it'll pick up again here and replicate that area. Okay. All right. Go watch some videos. It's like way easier to understand. Go watch videos on DNA replication. There's really good ones on mastering biology, and that I linked to the thing link. Okay, go do it. Go, get out of here.